Hello, fellow biologists. Welcome to our next lecture on plants at the organismal level. I know that I've had a little trouble in the past with the recording, so I've got my very stylish microphone here. I hope that works for making the volume a little bit better. And I also am working with a laptop that I know it doesn't have the greatest camera, so my office at the moment looks very yellow, but it's not actually that yellow. You just got to take my word for it. I'm not really living in a yellow submarine. It just looks like that. All right, so where are we? In our previous lecture, we had talked about plant structure. And in particular, we talked about organs, we talked about tissue systems, and we talked about cells that make up those tissue systems. In this lecture, I'm going to start talking about plant development. In other words, how do these organs, tissues, tissue systems, etc., change over time so that the plant can solve the problems that it faces. And remember that the big picture that we're thinking about in all these lectures is how plants solve the problems of being a multicellular organism. How do plants solve the problem of gathering resources? How do plants solve the problem of reproduction? How do plants solve the problem of avoiding predators? And then eventually we'll think about, at the physiological level, how plants solve the problems of maintaining an internal environment. How do they maintain, or how do they achieve gas exchange? How do they achieve circulation, and so on. We're going to talk about plant development. And I'm going to sort of think about this in two big sections, meristems and morphogenesis, or the M&Ms of plant development. And so meristems are a concept that you were introduced to in the previous lecture about plant structure, but I'll talk a little bit more about meristems and how they work to create our new organs. And then we'll talk about morphogenesis. And so there are different definitions of morphogenesis. I think in an animal world, morphogenesis has a somewhat narrower definition. Morphogenesis refers more precisely or more narrowly to development of a particular organ, but since plants are collections of organs, and these organs are constantly being produced over the course of a plant's life, I think of plant morphogenesis in a little bit of a broader concept for plants, right? So they're not just the forms and the types of the organs, but they're also sort of how plants achieve their particular body structure, their particular body plan. Right? And so I'll break morphogenesis into three parts. We'll think about gene expression that's involved in morphogenesis. We'll think about how gene expression then is influenced by growth regulators, such as plant hormones. And eventually we'll think about how environmental cues influence the growth regulators, which in turn impact gene expression. And so this is going to span a couple of chapters. So meristems and gene expression are in chapters 35, and growth regulators and environmental stimuli are in chapters 39. And I think they go together very well, but apparently I disagree with Campbell. The big picture, remember, is that plants need to solve problems, right? And so what I'm going to argue is that plants behave. And I'm in great company with this. The great Sir David Attenborough once started an entire book on plants with the sentence, "Plants behave." All right? So plants don't necessarily behave in the same way animals do, in the sense that animals often, their behavior is mediated through the movement of particular organs, and the movement of organs is often achieved by movements of muscle cells and so on. So plants aren't going to behave like that, but plants are going to develop, and that's the way they behave. That's the way they respond to many of these problems they encounter in their external environment. Plants don't necessarily behave on quite the same time scale that we're used to thinking of animals behaving. So we move relatively quickly. Other animals like little flies and insects can move even faster. But plants behave at a somewhat different time scale. And so we often aren't aware of plants behaving. But they really do. And so to convince you that plants behave, what I'd like to do is I'd like to show you just a few videos of plants in motion, plants moving. And these videos were actually produced, they're time-lapse photography, they're produced by one of my committee members, former committee members in graduate school, Dr. Roger Hangarter at Indiana University. The videos I'm going to show are what are known as examples of tropisms, and so these are developments of plants in response to and towards a particular stimulus. And so light, of course, is a great stimulus. So to set up this video, we have a little light growing above a series of little corn coleoptiles. So these are little corn shoots, a little bit younger than the ones that you saw in lab. And what we're looking at is a series of time-lapse images taken in about 10-minute intervals over 18 hours. And so the light is going to be on for a number of hours, and then we're going to switch this very directional light to a much more diffuse overhead light and see what happens. And you can see why this video is titled Worshipping the Light. So over time, the little coleoptiles are going to start to grow towards this very directional light source. But in just a moment, we're going to change where the light is coming from. And now an overhead light is being used, and notice that as our overhead light is being used, our little coleoptiles are growing instead of towards the specific light back up towards the regular light. However, they were doing this by growing and by developing. So notice that there's there are little signs over here that they had been bending towards the light as they're now growing back up. All right? So in development, we can often see a history of how plants have responded to the environment in the past. Shows a similar behavior and tropism in plants, but this one is natural in the sense that it actually occurs in a real environment instead of the artificial lab environment. And this is a sunflower, and we're going to see what our sunflower is doing over the course of a day as the sun passes from one side to the other of this particular field. So notice that as the sun is overhead, our sunflower is going to open up its leaves to catch all the light of the overhead sun. But as our sun starts to set, notice that our leaves are going to point towards the setting sun. And then as the sun goes down, our leaves sort of collapse and sort of take on this position that they would overnight. But even before the sun starts, notice that our sunflower is starting to catch the first rays of the next day, and it's moving its leaves back into position to do that. So this is sunflower behavior over the course of a particular day. Another type of response are what are known as circadian responses. And so these are responses that occur over the course of a day, over the course of a particular 24-hour period. And there are lots of plants that indicate a variety of circadian responses, but since we're so close to Halloween, I thought it would be fun to show a circadian response of a developing fruit, particularly of a developing pumpkin. So imagine, of course, that you're Charlie Brown in the Great Pumpkin Patch. And so this is what the Great Pumpkin is going to look like as it grows over the course of a fall. So here we see the daytime, right? This is the night, and notice how much our pumpkin is growing in the night compared to how much it's growing in the day. So basically our pumpkin sort of inflates during the night. You can see it go here. But in the day, it doesn't grow that much. It inflates in the night, not so much in the day, and so on. And so this is a pattern that's indicative of a lot of fruits. A lot of fruits are actually expanding much more during the night than they are during the day. And lots of carbohydrates during the night are being pumped by a phloem into these giant carbon sinks. And so we'll talk a little bit more about that eventually as we talk about the movement of carbohydrates. But I wanted to also illustrate this as an example of circadian rhythm, daily movement of plants. So you can see plants behaving over the course of a day. Of movement or behavior that plants display are nastic movements. And so in a tropism, what a plant is doing is it's growing towards a particular stimulus, like a light or water or something like that, or maybe away from a stimulus such as gravity. But in a nastic movement, this is a behavior based on a stimulus, but the direction of the plant is not really growing towards a particular stimulus. And to illustrate this, we have several morning glory vines. Morning glory vines grow relatively quickly. They produce flowers often overnight that appear in the morning, hence they're called morning glory. But because they're vines, they need to twine around something to hold themselves up. And as they go through their environment, what they're trying to do is they're trying to find some sort of structure like these poles that they can wrap around. So notice the way that morning glory vines search for a particular resource like a structure to hold on to. Again, this is time-lapse photography. But notice the very exaggerated movements over the course of just a few hours that these vines are displaying as they try to find particular structure to hang on to. And they're sort of creating these wide circles. They're getting wider and wider until they find something. And when they do, then notice that their patterns of movement and their patterns of growth change very substantially. So here's another one. It just found its pole. So notice that it's wrapping around its particular pole. This one found the pole over here. And now they've found a structure to hang on to, and all's well in their world. Some nastic movements or behaviors happen so rapidly that we can actually see them in our time scale without the use of time-lapse photography. This is what we're going to see for a plant that's known as the sensitive plant, the mimosa plant. And mimosas aren't just drinks, they're actually plants that you can find all over the tropics. And they're very common weeds, in fact, so common that you can just walk through a field and accidentally step on them. And so students who go to Belize sometimes do this, and when they step on these sensitive plants, the plants respond, often in a way that catches students a little bit by surprise and can be a bit frightening or spooky if you don't know what's happening. So here's the leaf of a sensitive plant, and notice that it's a doubly compound leaf. So here's a leaflet, here are many, many smaller leaflets over here. And what we're going to do is we're going to stress one of the leaves by putting a little flame under it. And so remember, this is not time-lapse photography. Look at how quickly our sensitive plant is going to respond to this particular stress of the flame. So there's the flame. And notice how quickly the leaf folds in an attempt to avoid whatever this particular stress might be. So this is probably a reaction against a potential herbivore attack or some kind of other stress that might challenge the leaf itself, and so the plant is going to respond by folding up the leaf. Last but not least is the Venus flytrap, the one you've all been waiting for. And so yes, these are also nastic movements, and these are nastic movements that of course happen fast enough that we can see them, and happen so fast that they are capable of trapping a fly. Our Venus flytrap has a leaf over here, so remember all plants have just 3 organs, and this particular organ is a leaf. They're little trichome hairs, so these are hairs produced by the epidermis, if you remember epidermal cells, and if a fly or something else triggers one of these particular trichome hairs, what's going to happen is that the leaf is going to close up quickly enough by bringing water into cells. It's going to bring lots of water into cells very rapidly and close up quickly enough to trap the fly. But it usually takes a couple of triggerings of these trichome hairs to make that happen. So here we go. Wham! And there it is. Okay, so hopefully you weren't a fly caught in the middle of it. I liked these videos of plants in motion, and it gives you a whole new appreciation for the way that plants behave. If you'd like to see more of these, you can go to plantsinmotion.bio.indiana.edu to see the work of Roger Hankarter and his lab with meristems. And as you may remember from our previous lecture, meristems are regions of undifferentiated cells that are dividing. And they divide to produce more cells behind them or to either side of them that will then eventually become differentiated to form the organs, the tissues, the particular cells that make up the plant body. We talked about meristems being in stems and meristems being in roots, and that's absolutely true. Let's also, as a quick reminder, think about where meristems occur specifically in these stems and roots. So most meristems that we talked about in our last lecture were what we referred to as apical meristems because they occurred at the tips of stems or would-be stems, or roots. So we have terminal meristems, and our terminal meristems would occur at the tip of a stem right here or at the tip of a branch. And we also talked about axial meristems, and axial meristems are those that occur at nodes. So we have terminal and axial meristems as two different types of apical meristems on stems. For roots, however, because we don't have nodes, we only have root tips. We just have axial meristems that are in effect terminal meristems. And we see those at the tips of these roots right here. In a little developing stem, and you saw this in lab, we can see these meristems in action. So for our little coleus shoot tip over here, this is the shoot apical meristem right here. This would be the terminal meristem. It's developing, it's dividing and producing more cells behind it that will eventually become our tissue systems, ground, vascular, and dermal. And these meristems over here that are growing right beneath the little primordial leaves are axial meristems. And so they're going to stay quiescent until eventually some kind of hormone signal causes them to start developing and producing branches. But right now it's the terminal meristem in a growing stem that's dividing to produce more of our tissue. These apical meristems, when they divide, produce what's known as primary growth, or the elongation of stems, the elongation of branches if an axial meristem divides, or the elongation of roots if a root apical meristem is dividing. However, a second type of meristem that we talked about are lateral meristems. And there are two different types of lateral meristems that contribute to what we call secondary growth, or the increasing of girth. So as stems get taller, right, they become a little bit more wobbly, as you saw in our plants in motion videos. So in addition to becoming taller, they're also going to have to become wider. And to make these plants, to make the stems wider, or to make the roots wider for that matter, we need lateral meristems. And in stems, we have a couple of lateral meristems. We have the vascular cambium and we have the cork cambium. So the vascular cambium, as I'll show you in just a minute, is going to produce our vascular tissues. It's going to produce xylem cells on the inside, and it's going to produce phloem cells to the outside. Meanwhile, our cork cambium is going to produce a particular type of dermal tissue. More specifically, it's going to produce periderm, and the particular type of periderm that it produces is going to be called cork, unsurprisingly. If this is our cross-section of a stem, a primary stem, we hopefully see structures that you guys recognize now. So notice that we have little bundles of vascular tissue, and this would be in a eudicot stem cross-section, as you guys saw in lab. And on the inside of this particular diagram is the xylem. So I'll put a little X over here for the xylem, and then we have a little phloem on the outside. And unfortunately, this picture flips the colors from what they often are, where xylem is usually depicted as red and phloem is usually depicted as blue. Our vascular cambium is going to occur between these two types of vascular tissue. So it's between our phloem and our xylem right here, but it's also this very thin layer of cells that occurs in a circle all the way around these bundles of vascular tissue. Now, as time passes, this vascular cambium is going to start to produce more xylem on the inside and more phloem on the outside, which is what we see in the second picture over here, which would be about a year later. In our second picture, we still have our initial bundle of xylem, we still have our initial bundle of phloem, but now we've added over here two new layers of vascular tissue. We've added some secondary xylem on the inside of our vascular cambium, which is this thick red line, and we've added some secondary phloem on the outside of our vascular cambium, which is indicated right here in red. This is an expansion of our vascular tissue, and this will continue every year for as long as the stem continues to expand. That gives the stem a lot of support, particularly from the xylem, but the stem also needs to protect itself from the external environment, and so we need to add dermal tissue as well. Particularly, we need to add periderm. This is what we see with our new region developing right here. So this is our old dermal layer right here, and just underneath the dermal layer we're going to produce the cork cambium. The cork cambium is going to start dividing to produce a new layer of cork, and so our cork layer is this layer right here. Fortunately, there aren't a whole lot of textbook figures that I could find that would go all the way from a primary stem through the start of secondary growth and eventually into what we'd recognize as a sort of very mature stem, a tree trunk. So I have done my best to produce a couple of pictures to illustrate what is going on, and I hope that these pictures will help. I've begun over here with a primary stem in year 1 that I think looks very much like this picture right here, in that we have vascular bundles with primary phloem, primary xylem in each bundle, and we have a vascular cambium that goes between the xylem and the phloem and that connects these bundles. Notice that we don't yet have a cork cambium. In our second year, we've added, our vascular cambium has divided, and we've added a second year of xylem. So this is our first year of primary xylem. We added a second year of phloem, and this is our first year of secondary phloem. Our primary xylem and phloem still haven't gone anywhere. They're right here in these particular bundles, but they're getting pushed apart by the secondary xylem and the secondary phloem. We also have had the formation of a cork cambium. So I've drawn a little cork cambium line here in black and started to produce some cork in brown outside of the cork cambium. So new cork cells are going to be produced external to the cork cambium. Let's fast forward a few more years. So if this is a tree that was planted when you first came to Georgetown, here we have a 5-year-old tree that you could come back to as a brand new alum. Notice that we've now added, we now have a total of 4 years worth of secondary xylem, 1, 2, 3, 4, produced in these rings. Here's the 5th year of primary xylem. And we also have a lot of secondary phloem. And so the secondary phloem is going to, instead of appearing in rings, is going to appear in these weird sort of pyramidal forms. And what's going on here is that our oldest phloem is out here, and our youngest secondary phloem is right here. And so when this old secondary phloem was produced, the diameter of our tree was a lot narrower than it is right now. And so as we've added more and more and more xylem rings, we've pushed our phloem further and further out, which means we've pushed our phloem apart. And so instead of our phloem, secondary phloem being connected, our secondary phloem has ripped apart into these little bits. And then the next year, our region of phloem here ripped into these 2 bits down here, and so on. And so this is giving us the pyramid shape that we see. If our youngest xylem is towards the outside, our youngest phloem is towards the inside. There is some cortex tissue right here, some ground tissue that's filling in between these pyramids of secondary phloem. And eventually this will disappear, but it's not quite gone a few years into development. If this is what our vascular cambium has been doing, our cork cambium has also been very busy. So our cork cambium produced a layer of cork out here, but much like our phloem, our layer of cork was ripped apart as our diameter expanded. And so I've tried to draw this with some of these jagged edges of our cork over here. And this is what you see when you look at the bark of a tree. That's often very rough, where the bark has been pulled apart. What that means is that not only have we ripped apart our cork, we've also ripped apart our cork cambium. So our cork cambium has to generate anew every year. So it generates sort of at the boundary over here between the phloem, secondary phloem, and the old cork. So it regenerates and it produces a new region of cork. And so you can see this has been going on for several years, although we started to lose a bunch of cork here that's just fallen off the outside of the tree. Right? So even though this tree is 5 years old, we probably only see like 2 or 3 years worth of cork growth right here. So this is our newest vascular cambium producing cork. Now, fast forward from our tree that's 5 years old to our tree that's 10 years old. So if this tree was planted when you first came to Georgetown, this might be a tree that you come back to see when you found that future special someone in your life, and maybe you even have a kid and you started a family. You're rich, you're famous, you can donate money to Georgetown Sciences to plant trees around campus. This is what that tree is going to look like now. So we have lots of rings of, specifically 9 plus the starter, 9 rings of secondary xylem that have accumulated. But notice that our phloem has basically been reduced to a single little ring over here. And that's because our phloem keeps getting crunched and keeps getting pushed away as our xylem accumulates. And so eventually we just got this single ring over here. We have a few rings of cork that are still left, but again, our cork keeps getting pushed out and it keeps ripping and falling off. So we only see a few layers of that. And so if you look at a tree trunk, what you're basically looking at is lots of secondary xylem, a very small ring of secondary phloem, and cork. And so that's how we go from a tree that is 1 year old, a seedling that's just starting to grow, to something that's starting to look pretty mature and pretty woody. I hope this series of pictures has helped you understand how a tree develops secondarily, right? How it expands its girth using the vascular cambium and the cork cambium. It provides a different view in that it combines these views sort of a two-dimensional cross-section, as we've just been looking at, with a three-dimensional view of a whole branch that also shows the longitudinal sections. And this is a very helpful view as well. So in this particular view, let's start at the tip of our stem over here. We can recognize our terminal apical meristem, and we can recognize some axial meristems on the stem during its first year of growth. You recognize, hopefully at this point, the vascular cambium, which is this red line right here. You recognize the primary xylem in blue, unfortunately, the primary phloem in sort of this brownish color, and the start of some periderm, some cork being produced right here by our cork cambium. And remember that then the vascular cambium is going to start to produce secondary xylem inside, secondary phloem to the outside, which is what we see here in year 2. So in year 1, in this particular cross-section, here we have our layers, primary xylem, vascular cambium, primary phloem. Time passes, and notice that our vascular cambium is sort of going to be pushed out, and it's going to start to produce more more xylem, now secondary xylem to the inside, and more phloem, now secondary phloem to the outside. So we see this accumulation of our first two secondary layers of xylem and phloem. As more time passes and we get to another year, we still haven't lost our primary xylem, we still haven't lost any of our layers of secondary xylem, but now we're adding more layers of secondary xylem, we're adding more layers of secondary phloem to the outside, and that's what we're seeing here. More secondary xylem, more secondary phloem, and the girth of our branch is expanding. And so as the girth of our branch expands, our cortex is going to our cortex is going to get pushed out. Our cork cambium is going to keep producing more layers of cork that are also going to get pushed out. And so here, finally, we can see in this fine little region right here, we can see the expansion of our cork produced by the cork cambium, but this is never going to get fairly large because, as you can see, our cork is continually falling off as the girth expands. The last term that I'd like to mention in this diagram is bark. And so bark is often considered to be cork, but it's more than cork. In fact, it's everything that's exterior to the vascular cambium. So in this particular picture right here, here's our vascular cambium right here in red. And so our bark is everything that's external to that. So it's the secondary phloem, any primary phloem that's left over, it's our cork cambium, and it's any cork that's still remaining on the outside of the tree. So there's actually a lot in bark. There are living parts of bark, there are non-living parts of bark, and these very different parts of bark are going to be really important, as we'll see in just a second when we think about how trees function. Just as a reminder, as secondary growth continues, a xylem accumulates on the inside, phloem accumulates on the outside, although it's getting crunched, and eventually then excess phloem and cork is going to be sloughed off, and the lost cork is going to be replaced by the cork cambium. At this point, I hope you'll be able to recognize our cross-sections of primary growth and our cross-section of secondary growth that you saw in lab. So this is a cross-section of a eudicot stem, a sunflower stem that you saw in lab, and you can see the vascular bundles right here, the xylem and the phloem. Note the different sizes of the xylem cells corresponding to the vessel elements that are wider and the tracheids that are smaller. On the outside of a vascular bundle, we have sclerenchyma fibers. And if you remember from our last lecture, sclerenchyma cells are differentiated cells. They're dead at maturity, and somewhat like xylem, they have these thick lignified secondary cell walls that also lend a little bit of support to our growing stem. This is a sunflower. Our picture of a secondary stem over here is not a sunflower. Sunflowers don't live that long. But this would be a picture of a tree, and I think in lab they used a basswood tree. So notice that in our little blown up picture of the cross section here, we now have several layers of secondary xylem. Again, notice the different sizes in the xylem cells. The larger cells are vessel elements, the smaller cells are tracheids. The vessel elements tend to be produced earlier in the spring when there's more water, fewer of them produced later in the summer and fall when it's less wet. This is our vascular cambium right here that's producing the xylem cells on the inside, the phloem cells on the outside. Here we see our wedge of secondary xylem, so several wedges of secondary xylem. And this is our cork cambium that's producing more layers of cork to the outside. And all of these layers from the vascular cambium out constitute, and by thinking about what the difference is between cork and bark. So here are a couple of questions that I hope will illustrate that difference. The first one is, let's think about cork. And so cork that you would see in the top of a wine bottle or bourbon bottle maybe is actually the cork, the outer layer harvested from trees. And in particular, there's a species of oak, Quercus suberii, that grows commonly in the Mediterranean, Portugal, Spain, etc., that produces a lot of the cork that is used for drinks, as stoppers for drinks. So as we think about cork, what tissue is this? What cell type? And remember that the tissue, the broad tissue group is the dermal tissue group, specifically it's periderm. And of course, we're talking about a type of periderm called cork that's dead at maturity. It provides a nice barrier. It keeps water from escaping from the tree trunk, from the xylem and the phloem. But of course, it also keeps various threats at bay, keeps a lot of insects out and other dangers from the tree. The second question, though, is about bark. So let's imagine girdling a tree. And so the idea behind girdling a tree is that instead of taking just the cork tissue, we take all of the bark in a strip all the way around the tree. Now, harvesting cork doesn't kill a tree because, of course, these workers right here are very skilled workers. They're just trying to take the cork. They're not trying to take all the bark because they don't want to kill these trees. They want to keep these trees growing so they can keep harvesting cork. So girdling a tree removes all of the bark, and eventually this will kill the tree. So why does removing the bark kill a tree, but removing the cork doesn't? And to understand this question, you have to remember that what's going on is that bark is everything external to the vascular cambium. So if we take all of the bark, what we're doing is we're taking the phloem as well as the cork. And so if we have a little region of phloem here that's missing, what that means is that the leaves at the top of the tree that are photosynthesizing can't get their carbohydrates past that break down to the roots. And of course, that means the roots die. And so once the roots die, then the roots can't send water up the xylem to the leaves, and so eventually the rest of the tree dies as well. So there's a big difference between cork, which is the outer layer of bark, and all of the bark, which includes both the living and the non-living parts, and the living part includes phloem, which is essential for transporting carbohydrates. We understand what meristems plants have, and that we understand that these meristems are producing new cells. Our next question becomes, how do these new cells that are produced develop into the specific tissues and eventually into the specific organs that we have in a plant? And there are several influences on these cells that cause them to develop in particular ways. So certainly we can think about how genes are expressed inside these cells that cause them to take on particular differentiated forms. It turns out, though, that the genes that are expressed can be influenced by a number of plant growth regulators, including plant hormones. And that in turn, these plant growth regulators can be influenced by a variety of environmental cues or other environmental factors. And so gene expression and eventually the form that a particular organ takes is not just a function of the genes that a plant has, but also function very much of the environment in which the plant is growing. Our broad study of how particular organs develop, and thus how the entire plant body develops, is a process known as morphogenesis. And so in the second section of the lecture, I'm going to explore morphogenesis and particularly explore this process of gene expression influenced by plant growth regulators, which are in turn influenced by environmental cues. Start with gene expression. And as you remember from comments that Dr. Stevens made at the very start of the semester, all cells in multicellular organisms have the same genes. Right? So all cells in our body, for example, with occasional exception, but basically all the cells in our body have the same sets of genes. However, our cells take on various forms. And so the reason they take on different forms is because they have differences in gene expression. So the genes that get expressed in a muscle cell are different than the genes that get expressed in a nerve cell in your brain, and so on. What that means is that many of the genes that regulate development are transcription factors. They regulate these gene expression, and by gene expression we're talking about the transcription of a gene from DNA to messenger RNA, and then the translation of the gene from messenger RNA to proteins. So these genes that we're going to be looking at that regulate development are going to be usually transcription factors that set off these cascades of gene expression. As we think about gene expression, we're going to think about how gene expression can impact the development in an organism in three ways. First of all, in terms of the number and the placement of organs. Right? So for example, we think about the fact that we have a couple of arms and a couple of legs. Well, sure, right? The number and the placement of our organs is very precisely determined. We're also going to think about the form of the organs, right? So, how long are our arms, right? How long are the various bones in our arms? And finally, in addition to thinking about the form, we're going to think about the size of cells within each of these organs. And size of cells also contributes to the size of the organs. Let's begin with number and placement of organs. In animals, genes that often control number and placement are homeotic or Hox genes. So this is an example of how a Hox gene would work, and we can see this in a mutant known as the antennapedia mutant of a fruit fly. So here is the head of a fruit fly, and you can see its eyes and its mouth parts and its antennae. And its antennae are actually, as we'll learn in our animal section, jointed appendages. Now, if we have a particular mutation that's known as the antennapedia mutation, that's a type of Hox gene, instead of getting antennae, where the antennae are normally found, we would get legs, right? That's the ped part of the antennapedia mutation. So these are some somewhat abnormal looking, but nonetheless fruit fly legs coming out of its head. So very scary. If any of you are looking for a really cool Halloween costume with a biological theme, you can get a fly mask, paste a couple of legs coming out of it, and you can be an antennapedia mutant. Plants, the number and placement of organs isn't usually regulated by Hox genes. Instead, it's regulated by a family of genes that we refer to as box genes. And one example of a MADS box gene in plants, actually 3 MADS box genes, would be the meristem identity genes that determine when a meristem will actually develop as a flower. So a meristem, remember, is a series of cells that are undifferentiated and dividing to produce more stems, leaves, and roots. But when a flower forms, what happens is that meristem actually becomes a series of differentiated cells, and these cells are differentiated into the cells that are going to become the flower. And remember that flowers have 4 whorls, right? They have sepals, they have petals, they have stamens, and they have carpels. So it turns out that there are 3 meristem identity genes that seem to control the formation of these 4 whorls. So these genes are labeled very creatively A, B, and C. So this is known then as the ABC hypothesis. As our meristem first starts to undergo this transition from being undifferentiated cells to differentiated cells, the first gene to be expressed in this sequence of ABC genes is the A gene. And so when the A gene is expressed, we have sepals. So just the A gives us sepals. Now, the A gene is going to continue to be expressed, but as the A gene is expressed, the B gene is going to turn on too. And so when the A gene and the B gene are turned on, we get the formation of a new whorl, and that would be the whorl of petals. Eventually, a third gene is going to turn on, and that gene is the C gene, and the C gene is going to inhibit the A gene. So the B gene is going to keep being expressed, but now we have the C gene instead of the A gene, and so when we have the B and the C gene expressed, instead of getting petals, we get stamens. And finally, the B gene is no longer going to be expressed. We only have the C gene left, and when only the C gene is being expressed, that gives us carpels. These genes was first identified through a series of mutants in which these three genes were turned off. And once we turn off each of these genes, we get a very different appearance of whorls, and so we can figure out what role each of these genes plays. Let's start again with what's known as our wild type plant that has our normal four whorls. And so if this is a top view, so to speak, this is going to be a side view. And so in our wild type plant, we're going to first have the A gene being expressed. And when just the A gene is expressed, remember that gives us sepals. The A gene is going to continue to be expressed through time, but the B gene is also going to be expressed. So when the A and the B gene are expressed, that gives us sepals. The B gene is going to continue to be expressed, but a C gene is going to be expressed and inhibit the A gene. And so when B and C are expressed, we get stamens. And finally, the B gene is no longer going to be expressed. We're just going to have the C gene. And when we just have the C gene, that's going to give us carpels. So what's going to happen when we lack the A gene? And so when we lack the A gene, we don't have anything to inhibit C. So C is going to start. And when C starts, that's going to give us a carpel. C is going to continue. B is going to appear. So now we get a stamen. C is going to continue. B is still going to be there. So we get more stamens, and eventually B stops being expressed, but C is still there, so we wind up with carpels. Notice that we've completely done away now with our with our sepals and with our petals. If B is lacking, then we're going to start out with A, which is going to give us sepals as it normally would. We're going to continue with A, but no B is there, so we get more sepals. Then eventually A is inhibited by C, and when C is present, we get carpels, but there's no B. And finally, we have more C that gives us more carpels. So in this case, what we do is we completely lack anything produced by our B genes, so we lack the petals and we lack the stamens. Finally, if we don't have a functioning C gene, then what we have are sepals, petals, more petals, and more sepals. In other words, we have no reproductive whorls. What I'd like to make in all of this is that the microenvironment is very important. Notice that what gene gets expressed is often determined by the surrounding cells and the surrounding whorls. So if we have the A gene being expressed initially, that determines what gene is going to be expressed in this next group of cells that will eventually develop into petals, and so on. So microenvironment, the developmental environment, the surrounding cells, impacts in a very important way how each subsequent cell is going to develop. The way that gene expression can impact development is by determining the form of organs. And there are lots of genes that determine forms of organs in plants. One particular example that your textbook mentioned is the gene knotted, knotted1. And knotted1 determines how finely a leaf is divided. So here's a typical tomato leaf, and notice that our tomato leaf is compound, once, maybe twice, depending on where we're looking. But if we overstimulate knotted, what that means is we're going to make our tomato leaf that just is hyper compounded. We're going to divide our blade again. So this leaf, sort of difficult to tell, would be compounded at least 4 or 5 times. And so this is an example of a gene that can determine the form of an organ. And here's one other example over here in Arabidopsis. So here is a normal simple leaf, but when knotted is overexpressed, notice how our normal Arabidopsis leaf becomes highly compound. I also like to point out that environment is very important in the form of organs too. And we see this over here in the timing of phase change. So as a plant starts to grow, sometimes it will begin growth in what's known as a developmental phase. Sorry, in a juvenile phase. And the juvenile phase can often have organs, such as leaves, that have a very different morphology. So this is a little acacia seedling starting to grow. And notice that its leaves initially are very, very compounded, like compounded at least a couple of times. But as our acacia enters what would be known as its adult phase, when it's capable of reproducing, the leaves go from being multiply compound to basically being these broad, simple leaves. And the environment can determine when a plant makes this transition from a juvenile phase to an adult phase. So notice that organ form that we get, the form of the leaf, can be impacted by the environment and can determine which genes are expressed to make the leaf have a particular form, either not compounded or very compounded. Finally, gene expression can determine the size of the cells. And I won't talk a lot about this other than just to give an example of how cell size might be determined by something that's under genetic control, such as the placement of cellulose around the cell wall. And if plants place these cellulose microfibers around a cell wall in a circular pattern, but around the circumference, but not a circular pattern that would be sort of more longitudinal, what that does is it allows a cell to expand vertically, but not expand horizontally. And of course, this is what many plants are trying to do as they grow up, is expand vertically. So this would be an example of how something that would be under genetic control could determine the size of a particular cell and the growth direction of that particular cell. End this section on genes by pointing out that in all of these responses, the activation of genes is strongly influenced in some way by the environment. Maybe it's the external environment, maybe it's the internal microenvironment of the surrounding cell, but in one way or another, it's very influenced by the environment. And so what that means is that plants tend to be a lot more developmentally plastic than animals. Now, if we saw a horse, for example, with 8 legs, or if we saw a human with 8 arms, that would strike us as being very, very odd. That would be an example of developmental plasticity that we're just not used to seeing. And so occasionally humans have played around with this idea of what would it be like if we were more developmentally plastic. So here, for example, is our Hindu goddess Durga. She's a warrior goddess. She has 8 arms. Notice she has lots of weapons in her arms. She rides around on a tiger. That's one example. A little bit closer to home, maybe, is the Spider-Man character, Dr. Octopus, who gives himself, of course, 8 arms instead of his traditional 2. And, we're very intrigued by these figures and consider them as gods or as interesting action heroes. But what we don't realize, I think, is that we have plants all around us that are doing exactly this all the time. So here are 2 maple trees. One is in a forest, and you can see that in the forest we have very few branches, and on these branches are very few leaves. Here's a maple tree that's not growing in a forest. It's growing in a very open area. And look how many more branches it has. And of course, if you're seeing in the summer, it would have far, far more leaves. So, wow, right? Plants are sort of like our gods or like our superheroes, right? They can develop all these extra organs to do whatever it is they need to do. And so we just have to appreciate sometimes that we're surrounded by these godlike or superhero-like creatures that are behaving in the world all around us. Next, we need to think what determines which genes are expressed. And often what determines which genes are expressed are cell signals. And cell signals can take a variety of forms, but in plants we're going to call this group of molecules that can act as cell signals, we're going to call these plant growth regulators. And so these are signaling molecules of various kinds. They can be hormones, and the idea behind a hormone is this is a molecule that is primarily responsible for signaling. It doesn't have a lot of other functions. But there can also be a lot of molecules in plants that have a variety of other functions, such as glucose, but they can, as a secondary or other function, be responsible for signaling. So we're going to take a look specifically at just a few hormones, primarily just to give you an example of what some hormones do and how hormones can work together to influence gene expression. Your textbook has a list of many plant hormones, but this is by no means all. It thinks about where these hormones are found and the major functions. I'm not going to go through all of these. I'm just going to try to hit a few highlights, again, just to give you an example of what hormones can do and how hormones can work together. First, plant hormones I'd like to consider are plant hormones that promote cell growth. And I like to emphasize growth. So by growth, mean specifically the elongation of existing cells, as opposed to the division of cells and elongation as well. So auxin is a very important hormone that stimulates cell growth and is sometimes known as IAA. Its location is in the apical meristems of stems, and it can be produced by light. It also can be produced occasionally by seeds, but it's really this first location that controls a lot of plant development. And so auxin is, as I said, responsible for cell elongation in developing stems. And interestingly, it was Charles Darwin and his son that first did some of the initial experiments to demonstrate that there must be some sort of hormone coming from the apical meristem of a stem that impacts cell growth. So this is a very classic experiment that they did, and you can try a version of this if you want at home. What they have a series of little seedlings. These could be little bean seedlings, for example. And so an initial seedling, what they did was they cut off the apical meristem, and they shone light on just one side of the seed. Now, if this were a normal seed, as you remember from our plants in motion video, if this were a normal seed, the seedling should start to grow towards the light. But the Darwins noticed that when they chopped off the apical meristem, it just grew straight up, almost as if it wasn't responding developmentally to that light. To see what it was about the meristem that was causing this response, they tried a couple of subsequent experiments. So in the first one, they put this cap over the meristem so that the cap couldn't get any light. And once again, the seedling didn't bend towards the light, it just continued to grow straight up. But when their cap was translucent and the light could get through, sure enough, the seedling started to bend, which was also the case even when they put a translucent cap lower down. And so this led the Darwins to hypothesize that there was some sort of compound that was being produced in the apical meristem that was being transmitted further down the seedling that was causing the cells on this back side, this non-illuminated side of the seedling, to get bigger. And as these cells on the back side got bigger than the cells on the front side, these bigger back cells were pushing the whole seedling towards the light. You can see how apical meristems not only cause the direction of shoot growth, but also can impact shoot growth of other axillary meristems. So imagine we have a plant here with an apical meristem, and if we leave this apical meristem intact, it's going to keep producing auxin, and this auxin is going to suppress the growth of subsequent axial meristems below it. However, if we chop off the apical terminal meristem, then notice that these other meristems in the axils start growing, right, producing branches of their own, and the plant can suddenly go from being kind of tall and spindly to being very, very bushy. So that's what we talk about when we're talking about apical dominance, right? It's the ability of a terminal apical meristem to dominate or suppress the growth of other meristems along the length of a plant. And so auxin is responsible for this as well. Also, auxin can impact root growth, particularly the root elongation, and it can stimulate fruit growth as well. Again, meaning the expansion of cells in fruits. Second hormone that stimulates cell growth are the gibberellins, and I'm not going to say a whole lot about gibberellins other than often gibberellins can work in concert with the auxins. Another class of plant hormones promotes cell division and differentiation as opposed to cell growth. So if a cell gets bigger, eventually it's going to have to divide, and that's what this next hormone does, called cytokinin. And so cyto refers to cell, kinin is movement or division. If you remember from mitosis, cytokinesis is the final step of mitosis in which the cell divides and the two new cells with their new nuclei each go in their own direction. Plants, that's what cytokines promote. They're located in the roots as opposed to in the stems, and the apical meristems in particular, of the shoots. And so cytokinins are going to work in conjunction with auxins, but they're sort of going to be part of this tug of war, sort of this push and pull between cells growing and cells dividing. And it's often the ratios of auxins and cytokinins that are very important in plant development. So they stimulate the cell division in stems and roots. They can moderate apical dominance. So if more auxin increases apical dominance, more cytokinins lessen apical dominance. And finally, they stimulate seed germination, in contrast to what auxins do, which is to stimulate the growth of cells in the fruit. With the group of plant hormones actually inhibits growth and development. And so these are going to be sort of working usually opposite the role of auxins and cytokinins. We have abscisic acid, often abbreviated as ABA, and it's located throughout the plant in all organs. It inhibits growth, and it can do things such as promote the closure of stomata, the shutting down of these little holes, and the senescence of leaves as well, sort of the programmed death of leaves. And it can also inhibit seed growth. In other words, it promotes the dormancy of the seed. So an example of what this might look like in reverse, when we have low concentrations of abscisic acid, can be seen in mangrove seeds. So mangroves are trees that grow in the tropics. They grow in salt water, interestingly, and so their seeds are dispersed by waves. What happens in mangroves is their seeds usually start to germinate before they've even come off the tree. So here we see a couple of germinating mangrove seeds. This one is much farther along than this particular seed, but this is actually very advantageous for the mangroves because mangroves need sort of this little bit of a initial root to stick down, and so as the seed floats along in the water, if it brushes up against some sand, it will get stuck, and then the root will continue to grow and anchor the seed, and the mangrove can start to develop in that particular location. And so this early germination of the seedling is achieved by low abscisic acid concentrations in the seed. Another type of inhibitory growth regulator is ethylene. Again, it appears in all leaf organs, and it has a couple of effects. One is fruit ripening. So if you think about a fruit that you often get at the grocery store or in the calf, when this fruit is picked and shipped, it's initially not quite ripe to avoid damaging it in transit, but to sort of speed up the ripening, what sometimes happens is that they expose the fruit along the way in transit to ethylene, sort of to make the fruit ripen after it has been picked a little bit. And finally, we're surrounded right now by the effects of ethylene because ethylene also promotes the abscission of organs such as leaves. And so as leaves at this time of year lose their chlorophyll, and as the various nutrients get moved from the leaf back into the stem, eventually the plant wants to drop the leaf that's no longer going to be a useful organ over the winter, and the hormone that promotes the little break of the petiole from the stem is ethylene. What about how gene expression leads to the formation of organs? We've thought about how plant hormones can act as signals that stimulate gene expression. And so the last thing that we need to do is think about how environmental signals can stimulate our plant growth regulators such as hormones, which in turn can impact gene expression. And plants respond to a lot of different environmental signals, such as the length of light during the day, the quality or the quantity of light, quality being the wavelength, quantity being the intensity of the light, in temperature, of course. Even a variety of touches, mechanical touches from water or wind can be important environmental signals as well. So let me show you a few examples of some common and very important environmental signals that are involved in plant development. So the first one is photoperiod, or the length of the day. And so each day is divided into 24-hour periods, and the photoperiod is often thought of as the number of hours of light during that 24 hours. And plants can respond to photoperiod in a couple of ways. Some plants are known as short day plants, or SD plants. And the idea behind a short day plant is that it goes to its next stage of development, often the production of flowers or fruits, when the length of the days become shorter. However, as it turns out, we're going to see something a little bit interesting about day length. So let's start with a classic qualitative short day plant, which would be cocklebur. And so cocklebur grows throughout the summer, and you can see it growing right here. It doesn't flower. So in the middle of the summer, there might be very long periods of day length, say 16 hours, and a short night period. So it's not flowering. However, as we go from the summer to the fall, what happens is our day lengths lessen, we have longer night lengths, and our cocklebur then starts to produce flowers and eventually fruits, which are these very spiky little burrs right here. So the question is, what is actually stimulating the production of flowers and fruits? Is it the length of the day, or is it the length of the night? And to figure out which it is, let's take a long night over here and let's interrupt it by a little burst of light. And it turns out that when we interrupt our night with a little burst of light, it's just the same as if we had had a long day. So what this tells us is that these short day plants are not actually responding to the short day, they're really responding to the long night. In other words, development proceeds when night length exceeds some sort of threshold value. And cockleburs are really cool plants, as I'll show you in just a second. All right, if we have short day plants, then a second group of plants, a second photoperiod response would be a long day response. And so the idea in a long day response is that plants go to their next stage of development when the day is long. And so in this plant over here, if we start, this is a henbane, and so it starts growing in the spring, early spring, when the days tend to be much shorter and the nights tend to be much longer. However, by the middle of the summer now, when the days are much longer, this stimulates our henbane plant to produce flowers. And once again, we see that if we interrupt our night, what happens is it's almost as if we had a long day. Once again, our long day plants, much like our short day plants, are not keeping track of the day length, they're keeping track of the night length. But now they respond when the night length is below a certain threshold value. So there are a lot of plants out there that have either short day or long day responses. And interestingly enough, there are a lot of animals that have short day and long day responses as well. So even think about maybe some common house pets you might know. If you have a cat or a dog and their fur gets a little bit thicker in the winter, or if you think about hares or particular species of bears or foxes, for example, that have fur that change color in the winter and then back again in the summer, these animals are responding to day length cues as well. So day length cues are very, very common cues, not just in plants, but in animals. And so ultimately what you can do is, if you're crazy enough, you can actually manipulate plants or other organisms to go through developmental changes when you want them to, and to figure out how that impacts their fitness and their ability to spread and increase their geographic range, like this particular guy was doing right here in the Upper Peninsula of Michigan way too many years ago. A set of examples pertaining to environmental signals fall into the category of photomorphogenesis. So this is morphogenesis, right? The formation and development of organs that are come in response to light, but not the length of light that we have in a day, but the responses to either light wavelengths or to light quantities. So let's start with light wavelength. Once again, let's come back to our trees over here. So if you are a little tree seedling, right here, and you are growing in a forest, you have to figure out whether you want to produce lots of stem over here and few branches, or whether you want to produce many branches coming off of your main stem. And if you're in a forest, clearly the idea is you want to grow tall as fast as possible so that you can get to the light, because there's not a lot of light underneath the canopy. Well, so how do you know, though, that you're underneath the canopy? And plants can actually detect the wavelength of light. So if there is a lot of green light that is coming through the canopy, this green light will actually cause plants to grow very spindly over here with very few branches. On the other hand, if we have a more full spectrum of light, as this particular tree is experiencing over here, it will become much branchier. So we see this as we walk around the world all the time, particularly as we look at trees on campus, they're very branchy. But even if you have a garden, you can see this as well, and some of you might have seen examples of this in a Bio214 experiment with bean seedlings. So if you grow seedlings in the absence of light, or if you grow bean seedlings that sort of lack a full spectrum of light, what happens is they can become very tall and spindly as they grow up, as their internodes elongate to try to reach the light. But if you grow bean seedlings under a full spectrum of light, then they will grow, they'll produce a lot more branches, they will be much more, the leaves will be much more green, they'll produce a lot more chlorophyll, and they'll be trying to capture the light that's available to them. This is an example of how an environmental signal, which would be the quality of light, can impact the expression of, can impact the production of plant hormones, things like auxins, gibberellins, cytokinins as well, to impact the expression of genes that determine how long cells get between their nodes. The amount of light can also be important, and the amount of light can sometimes determine characteristics of an organ, such as its thickness. So we see that over here with two sets of leaves of an oak. This is Quercus pagoda, and these leaves of a particular tree, notice that they are they're sort of on the outside, they get a lot more light, and they're much more deeply lobed. On the other hand, leaves that are produced in less light, maybe closer to the trunk, are sort of more rounded, not as deeply lobed, and so this morphology helps them capture lower quantities of light than these leaves would be experiencing on the tips of branches. So both light quality and light quantity are important in photomorphogenic responses. Finally, we'll end by thinking about some physical stimuli that might impact leaf development. And the variety of touches that I mentioned that are important for plant growth. And so for one particular plant, this is a water lily, the touch of water has an enormous impact on how its leaves develop. So the seedling of this water lily is going to germinate on the bottom of a little pond or river over here. And as the shoot starts to develop, the initial leaves are going to be very, very finely divided. In other words, there's a huge expression probably of genes such as the knotted 1 gene. And this seems to be due to a cue such as water, or maybe a lack of oxygen as well, impacting the leaves. And so as this water is moving past the leaves, as these leaves aren't getting a lot of oxygen, right, that's determining what plant hormones get produced, which are then regulating the expression of genes such as knotted 1. Eventually, though, when the meristem gets above water, it's outside of water over here, what it then starts to do is produce leaves with a very different morphology that is sort of much more rounded, not nearly as divided, and these rounded leaves collect light. They're better at photosynthesis. They kind of help the plant float on the surface of the water, and so this change in the physical environment, being underwater or being above water, is the environmental cue that sets along this pathway of changing plant growth regulators and gene expression. So I hope that you have enjoyed our trip through morphogenesis, from cues to regulators to gene expression. And I look forward to seeing you in the next lecture, when we're going to look at how plants actually regulate their internal environment instead of responding to external stimuli.